me. It's really an honor to be part of the conversation and possibly collaborations as we go forward. I'm going to switch my camera off so that it's not distracting anybody. So as mentioned by Sarah, that I will be talking about next generation sequencing to enhance the diagnosis and treatment of drug-resistant TB. And I think one of the lucky features of drug-resistant TB, why doesn't the page, I can't get it to move, sorry. Well, let's do it like that. One of the features of Mycobacterium tuberculosis, which may be different from other pathogens, is that resistance is in...
Professor Warren, we cannot see your presentation.
Oh, yeah. I see why. There we go. How about like this?
We still cannot.
Now you should be able to.
Yeah, we can't see.
Wonderful.
Yeah,
This shows you that I can also make mistakes. So, as I was about to say, I think that one of the points of tuberculosis is that the recombinants into the mycobacteria. It's really just a changing genome, which means that if we can understand that change in the genome, as shown very nicely in this picture, that this will give us the markers that we can use to identify susceptibilities or resistances to drugs. Currently, the diagnosis of drug resistance in most countries relies on either culture or a very smart lab in a box called Expert Ultra, which are which is a genetic-based test as well, based on the fact of these presence of mutations. And then there are certain line probe assays which allow us to discern additional information. But we are slowly moving towards a more comprehensive analysis rather than these very targeted approaches. One of the new tools that's just about to be released and evaluated and distributed all over the world is the new Expert MTB XDR cartridge, which is really looking at the second-line drug resistances. But again, that doesn't give us the full repertoire of resistance that we want to be able to discern. And so in the next two pictures here, you can see that there's whole genome sequencing, and we're showing a small mini-seq picture of the instrument. And then on the right-hand side is a deeplex kit, which has been developed by a company called GenoScream, which looks at a targeted approach, but looking at 13 different drugs and enables you to look very deeply into underlying populations. And so in this talk today, I'm really going to focus about whole genome sequencing, and I'm going to talk a little bit about targeted deep sequencing and how this can help us understand resistance and then use that knowledge to guide treatment. My talk is a bit of a potpourri of work that's ongoing in the lab. Some of it's not published, very few bits are actually published, but I thought it'd be nice to show how we're using this technology to understand the epidemiology of drug resistance and whether we can change the course of this epidemic. So if we look at whole genome sequencing, for those who are not familiar with the technique, we start off with DNA that's been extracted, and this is generally done from a culture. So there's always this delay in getting a specimen or isolate that is sufficiently clean. And that's really important to understand, that you don't want to sequence contaminating material. We then take that genomic DNA, break it up, add on linkers. We then link it to a flow cell, and very simply, then we allow the flow cell to create clusters, and those clusters are then sequenced, and we get data, lots and lots of data. So, and that data is short reads. And what one does then is aligns those short reads, as you can see in this picture, that there's a reference genome, and we show or use bioinformatics to align these relatively short reads along this reference genome. And we can sequence to about 100 times depth economically. If we want to go deeper, it costs a lot more. And then we look for, as I've indicated with X's, the presence of variants or mutations. And what one then wants to do is understand whether those variants are in genes that are known to confer resistance. Targeted deep sequencing is a very similar approach. We take genomic DNA, but in this case, we don't break it up. We PCR amplify. Again, we put on linkers, put it on sequence, but the output is very different. As you can see here, we have the reference genome, but we're only targeting one area. But we can sequence, in this case, to 100,000 times depth, which means we can find those underlying populations. And I'm going to show you in a couple of examples as we go along, which is quite important. And it's very different from what a whole genome is telling us, which is giving us the major population. So I want to say that, this is the technology that the WHO is now promoting. It's really trying to say, how do we move from the classical microbiology into an era of using next generation sequencing for surveillance, which I think we really do need, as well as can we guide therapy? And so I think in the next 5 years or so, this is going to build more and more momentum. I'll mention a little later that there's already a catalog of mutations in particular genes that are known to confer resistance, and there's various statistics associated with that. And that's going to be the catalog that we need for all of our bioinformatic analysis. Just trying to move on. So I wanted to start with this picture because I think that it really tells us about the epidemic. If we start with the little picture on the left, the little blue person, that person can start off with no TB and then get infected with a resistant strain. That person then can become infectious and they can spread, as shown in the middle of the picture. And then the people that are infected can go through this cycle again of becoming either latently infected or actively diseased and contribute to transmission. But next-gen sequencing can try to answer some of these questions. And I've written in red, as you can see. And by the way, all the slides will be made available to you. We can say to ourselves, can we use next-generation sequencing to identify ways of improving treatment? Can we find resistance before we would ever have found it microbiologically? Can we understand how resistance is evolving? Where are these resistant cases by using the fingerprints, if we want to call them, or the sequences of resistant isolates, and then mapping them onto maps and understanding where hotspots of transmission may be occurring? We can understand and maybe influence policy and guide healthcare resources to address the areas where. So I see that whole genome sequencing attacking this epidemic. I'm going to start with one or two, a couple of examples of studies that we're doing right now. The first one is a study that is trying to look at isoniazid monoresistance. Why are we interested in that? Because actually it's a hidden epidemic. If we're using the expert as our primary screening tool, it's only looking for rifampicin resistance. So we're actually ignoring all patients with isoniazid monoresistance unless their treatment and their clinician that's treating them has a suspicion that they are not doing well and that further investigations must be done. The other importance of this study is, contrary to many other studies which are being done in a metropole, this is a study done in a rural context. And my honest opinion is we don't do enough research in the rural context of Africa. Tend to focus on the big cities. And we need to really understand what's driving the epidemic in these rural environments. And the drug resistance surveys is that about 6% of patients have isoniazid monoresistance, meaning that during treatment, they have one less drug. And this is really important during the continuation phase when they're only being treated with two drugs, rifampicin and isoniazid. So in this study, what we try to do is we try to go to a number of clinics in a rural context of the Eastern Cape, and we wanted to collect all rifampicin susceptible isolates. We then sent those susceptible isolates to Stellenbosch University, where we did phenotyping using either 7H10 solid media or midget media. And then we used a number of molecular tools to try to identify the mutations conferring resistance. First, the more classical methods like the line probe assay or sequencing, and then we moved into the next generation sequencing. And I think from this study, we found things that we didn't expect, to be honest. And I think that's what makes the beauty of this. So we started with around 993 patients, and we did drug susceptibility testing on 766 of these that were culture positive. One lesson we learned is if cultures have to be flown from distant, we obviously lose viability. But what we saw was, in fact, that I'm wondering why my thing is not working. I'm just going to have to point it out to you. What we found is that 14% of the isolates that we received, this is at baseline, were resistant to isoniazid, much, much higher than what was being observed in the drug resistance survey. We then did this sort of standard test to look for what we call canonical mutations, or mutations known to had these canonical mutations. The rest of them didn't. That means if we use the standard method, we wouldn't be able to find this. So this was now quite different to what we expected. So then we said to ourselves, phenotypic resistance, and we could confirm resistance in around 20% of the isolates. And we went on, had what we called intermediate isoniazid monoresistance. So I'm sent, and here we could see, then we said to ourselves, maybe what we should do is we should sequence a bit more of the area of interest. But we only found 5 canonical mutations, really indicating to ourselves that even with targeted deep sequencing, which could look underneath the ones that didn't have mutations, the vast majority, 16 out of 18, didn't have classical mutations, probably implying that other mechanisms are involved. We then also looked at this intermediate group, which is classically classified as susceptible. And so I'm going to show you a little bit of the theory here. I'm just going to open the whole lot up. If you're doing a midget culture drug susceptibility test, you have a control that is a 1 in 100 dilution. And if your sample that's grown in the presence of drug grows before the control, it's considered as resistant. If it does not grow after the control, it's considered as susceptible. If it grows after the control, but before the cutoff of the assay, it's called intermediate. Now, classically, as I mentioned, this is called susceptible because it's growing after the control. So we now feel that this actually has meaning. It's not just susceptible. So what we did is we asked, maybe we should repeat this intermediate test on midget, but using the culture that had shown an intermediate phenotype. In other words, we'd enriched the population without drug, we'd enriched it with drug, and then if we repeated the test, would we now get a resistance classification? And is in fact resistant. So this means actually what we were starting with is a population that we could find a solid media to find that growth of the resistance strains in the presence of drug. We've then done targeted deep sequencing 15 hours in the genome, and we could explain these with classical mutations, but their underlying populations. So it's quite an interesting study because it's contrary to what we would have predicted. And if we look at the summary of the study, we're basically saying that 30% of isolates that are isoniazid monoresistance would have been detected by the standard assay. The other 60% wouldn't have been. That there are underlying populations that are not being detected because the tools that we're using are not sensitive enough. And there's another group that have resistances that are not actually in the target area of interest. So we really need to understand what is actually happening here. Why are we seeing these? Oh, no, sorry. Why are we seeing these differences to what we would classically understand? And one idea is this a consequence of IPT? I know I may be seen as a heretic to have to say that, but I think we need to really understand whether or not treatment of patients with isoniazid may be fueling this in the context of this rural environment. The next example that I want to move on to is also using targeted deep sequencing, and it's looking at this new anti-tuberculosis drug called bedaquiline. And this is a patient who was 48 years of age. He previously had TB when he was 12, HIV negative, and he was diagnosed with MDR TB. And when we look back, and I think this is a lesson for all, we started treatment based on the fact that the patient had rifampicin-resistant disease, and we started on a regimen, but we didn't know whether that regimen was appropriate or not. Unfortunately, we don't have the isolate at baseline in 2013, but we do have the subsequent isolates as shown with the red dots. And so the first one we could sequence was after the patient had been on treatment for a period of nearly 5 months. And what we could see very clearly, there was the rifampicin resistance, there was isoniazid low-level resistance, there was ethambutol resistance, there was ethionamide, there was fluoroquinolone, there was pyrazinamide resistance, and streptomycin. So when we actually look at the regimen, so in black, it's resistant. In green, it's susceptible. This poor patient was on only a 2-drug regimen, which is bound to fail. And so they changed the regimen. And they started a new regimen. We can quite nicely see that there were a few drugs that remained resistant. But there were, at least in the beginning, 5 drugs that were able to. They started then, after a few weeks, they included bedaquiline on compassionate grounds, and that patient was then treated. The injection led to some poor adherence or not wanting to take that, and one can understand that. So then we sequenced these genomes, and we started sequencing, and we found mutations conferring bedaquiline resistance after the bedaquiline stopped. But most surprisingly, when we started analyzing, we could see that resistance marker disappeared, and a new resistance marker appeared, and then it disappeared again, and another one appeared, until the patient finally had this black resistance marker, to make it simple. And the question really was, now we have, is this mutation, are these mutations actually conferring resistance? So we did the phenotypic drug testing. As you can see here, the patient was susceptible to bedaquiline, and there the resistance is. So therefore giving us confidence that these mutations were actually conferring resistance. The question was, did resistance actually emerge after the bedaquiline treatment was stopped or beforehand? And so we used targeted deep sequence, because then we could look earlier, and we could see after 2 months of bedaquiline treatment that there in fact were mutations. That mutation that became dominant after 7 months was already present. This is something we called pre-resistance, long before we would have detected it phenotypically, as you can see. It's quite important, because now this tool can allow us to find resistance very early that will allow us to manage the patient perhaps better. So what were we actually seeing in this patient? Did the patient have one lesion? And what was happening is that lesion then had another population growing out? Or in fact, did the patient have a different lesion? Each one multiple different populations. And my last slide, which is a bit later on, would really show you this as well. One of the interesting things, and maybe many of you have heard of this, that resistance is supposed to cause a fitness cost. But we also know that there are compensatory mutations that are able to ameliorate that resistance. And here we see in this patient how compensatory mutations known to occur in RPO-C are emerging. And then there's another mutation coming in RPO-B and another one in RPO-C as the population, different populations are present, implying that the bacteria is adapting itself to this new resistance mutation that it's got in order to retain its fitness and cause disease. The take-home messages from this study really are: treatment without knowing the drug susceptibility pattern raises the risk of treatment failure. We saw this in this patient, and it's a classical dogma that now we really need to address in all TB control programs. So we can't really just treat blindly. We can see that bedaquiline can emerge very rapidly, and it's a big worry because it's the only new drug. Well, now there are a few others in the last 40 years. But multiple different resistance clones are emerging. We don't quite know why. Is it because of different penetration of the drug in different areas of the lung? We don't know. That resistance actually emerged despite this patient taking 5 other companion drugs.